Hello, everyone. Welcome to another episode of Inflammatory Content, the podcast about immunology and biomedical research more broadly. For those that are new to the show, allow me to introduce myself and tell you what the show is all about. My name is Dr. Kellen Cavagnero, and I'm a postdoctoral fellow at UC San Diego with over 10 years of experience as an immunologist and biomedical scientist. This podcast has two formats. In some episodes, I do deep dives into the latest high-impact biomedical research findings. I call these Journal Club episodes. In other episodes, I have conversations with world-class researchers to broadly communicate the cutting-edge work going on in their laboratories and to get to know the people behind the science. So sit back, relax, and let's dive into some science. And if you enjoy the show, please consider sharing the episode, leaving a review, or donating to support my work. Thanks for listening. The title of our paper today is Spatial Proteomics Identifies JAK Inhibition as Treatment for a Lethal Skin Disease. The first author of this paper is Thierry Nordman, and the corresponding authors, there's three of them, are JAKi, Lars French, and Matthias Mann, and these authors are mostly from Germany. The article was published in Nature in October of 2024. I thought this paper would be interesting to share with everyone because these authors investigated a severe disease that lacks an effective treatment. They used two very innovative approaches, spatial proteomics and a high-throughput in vitro assay using patient samples. And these approaches led to the discovery of a new drug target for this severe disease that lacks an effective treatment. And importantly, there's already an FDA-approved drug that targets this molecule. Finally, the authors perform a clinical trial using this FDA-approved drug, repurposing it to treat this severe disease, and it is successful. All right, let's get into the background information. So what is this severe disease I keep referencing? Cutaneous adverse drug reactions. It's a class of diseases that includes maculopapular rash, or MPR, drug reaction with eosinophilia and systemic symptoms, or DRESS, Stevens-Johnson syndrome, or SJS, and toxic epidermal necrolysis, or TEN. TEN is a severe form of SJS, and there's also an intermediate form that is called SJS-TEN overlap. DRESS, SJS, and TEN are rare and life-threatening. All of these reactions are induced by drugs that would normally cause no problem in most people. Interestingly, non-steroidal anti-inflammatory drugs, or NSAIDs, and antibiotics are common causes of cutaneous adverse drug reactions. I'm going to provide a bit more background about TEN, toxic epidermal necrolysis, because this disease becomes the focus of the paper. A key feature of this condition is epidermal detachment, with greater than 30% of the body's surface area affected. The skin is literally falling off. Mechanistically, this is thought to be driven by CD8 T cell mediated keratinocyte cell death. What exactly is driving the CD8 T cells to do this is unclear, but it is thought that it is a combination of antigen-dependent CD8 T cell activation and bystander killing. Not only is the skin affected in this cutaneous adverse drug reaction, but there is also systemic involvement. These patients often have sepsis and multi-organ failure. This disease is extremely severe and it is fulminant, meaning it happens suddenly. The prognosis is grim. Greater than 1/3 of cases are fatal. The treatment: find and stop the culprit drug and manage the patient in the burn unit. There is no FDA-approved drug for this disease. There are a few experimental treatments: steroids, anti-TNF, and cyclosporin. Bottom line, this is a terrible disease and we need effective treatments. All right, now that we better understand the disease that is being studied, let's talk about the approach that the authors used to study it. Omics. Omics are unbiased approaches that have the potential to reveal pathogenic mechanisms. One type of omics approach is transcriptomics, which measures expression of all genes to infer cellular function. This is a popular omics approach because it is cost effective, easy to use, and we have well annotated reference genomes. Around the 2000s, bulk transcriptomics was developed. This allows you to measure gene expression in a sample, but it's the average of all of the different cells in there. It's like the blended smoothie. Around the 2010s, single cell transcriptomics was developed. This now allows us to look at the expression of every gene in every cell, enabling the discovery of new cell types and states. And most recently, in the 2020s, spatial transcriptomics was developed. Now we can look at the transcriptome of every single cell type in a sample and where it exists in space to better understand its function and role in disease. Transcriptomics is really great, very powerful approach. I use it all the time. However, RNA is not always translated to protein. Protein is a better measure of function than RNA. Enter proteomics. The paper we are going to discuss today uses spatial proteomics, which is a very up-and-coming field. There are two major classes of spatial proteomics approaches, and imaging-based approaches and mass spec-based approaches. There are pros and cons of each approach, but the main difference is that the mass spec-based approaches allow you to multiplex at a much higher level, thousands of proteins, whereas the imaging-based approaches only allow for dozens of proteins. The authors of this paper previously developed an approach called Deep Visual Proteomics, or DVP. This approach combines imaging and mass spec-based approaches. It starts with archived patient tissue samples, formalin-fixed paraffin-embedded tissues, which is important because it means we can leverage all of the samples we've historically collected. The tissues are first imaged and segmented using deep learning. Machine learning is then used to predict cell types. Lasers are then used to microdissect out individual cells. Finally, ultra-high sensitivity proteomics is run on these samples, yielding cell type resolved data. They did this for keratinocytes, the cells of the skin surface, and immune cells. Notably, for immune cells, they lumped all CD45 positive cells together. So they did not use the approach to resolve individual cell types of the immune system. The authors first showed the data from the keratinocytes. They detected around 4,000 different proteins in their samples. And they see by dimensionality reduction and unsupervised clustering, a disease-based clustering. We see the healthy samples looking different than the MPR, looking different than the DRESS, and looking different from the 10. Unbiased pathway analysis identified that the main difference between the different keratinocytes in these diseases was interferon response. These proteins involved in interferon response were highest in patients with TEN. So what are interferons? Interferons are a class of cytokines that are important for defense against viruses, but also play a role in some autoimmune conditions and cancer. In my recent work, I identified that interferons play a pathogenic role in the neutrophilic dermatosis sweet syndrome, but they have not been associated with these cutaneous adverse drug reactions before. When a cell recognizes interferons, the JAK-STAT pathway is activated that leads to the induction of interferon-stimulated genes. The JAK-STAT pathway is also important for the response to many other cytokines, and there are FDA-approved inhibitors of the JAK-STAT pathway that are currently being used for other inflammatory conditions. The authors next look at the CD45 positive immune cells. And like the keratinocytes, the authors see disease-based clustering of CD45 positive immune cells as well. And again, proteins related to interferon response were highly expressed in TEN. So to summarize so far, the authors have shown that the interferon response pathway is highly upregulated in TEN keratinocytes and TEN immune cells compared to other cutaneous adverse drug reactions in healthy controls. They then ask, which type of immune cells is this upregulated in? Now they're going to resolve individual cell types of the immune system. They separate out macrophages, helper T cells, and cytotoxic T cells. Remarkably, they were able to identify around 2,000 proteins using only 20 cells. What I would like to know, though, is how many proteins could they get with just one cell? Looking at these immune cell subtypes, they found that macrophages had the highest expression of interferon pathway proteins in TEN. Importantly, the authors validate this potent activation of the JAK-STAT pathway in TEN using more traditional approaches like immunofluorescence and immunohistochemistry, as well as more unbiased approaches like phosphoproteomics, which I'm sure was a huge undertaking, but surprisingly, the data were buried in the supplement for some reason. Okay, so that concludes the more descriptive part of the paper. Now the authors are going to address whether what they have found is significant. Specifically, they ask whether JAK inhibition prevents T cell killing of keratinocytes and ameliorates TEN. And they first approach this question using an autologous co-culture model and live cell imaging assay. In this assay, they take 2 samples from patients that have survived TEN. They take hair samples, and from these samples, they grow out primary keratinocytes, and they take blood. And from these blood samples, they isolate PBMCs, or peripheral blood mononuclear cells, which include the T cells. They then co-culture the T cells and the keratinocytes in the presence or absence of anti-CD3/28, which will activate the T cells, and in the presence or absence of JAK inhibitor that will block the interferon signaling. Importantly, a dye is also included in here called cell-tox green that binds DNA and can be therefore used to compute keratinocyte death. So what the authors find with this assay is that when the activated PBMCs are combined with the keratinocytes, there is a significant increase in keratinocyte death compared to when the keratinocytes are cultured with the resting PBMCs. However, when a JAK inhibitor is included in the culture, the killing is significantly suppressed. So JAK inhibition reduces T cell mediated keratinocyte death in vitro. One thing that's not clear to me is why the authors had to use samples from TEN survivors. I suspect that this assay would work the same with samples from healthy patients, since anti-CD3/28 is being used to activate the PBMCs. Moving on, the authors next ask whether JAK inhibition would work in mouse models of TEN. To address this, they use a mouse model of TEN induced by SMAC mimetic. SMAC stands for second mitochondria-derived activator of caspases. Intradermal injection of SMAC mimetic induces rapid detachment of the epidermis, just like in TEN. The authors find that when they pre-treat with oral JAK inhibitors, they can inhibit this epidermal detachment induced by SMAC mimetic challenge. Importantly, they also test out oral JAK inhibitors in a treatment model where they give the drug after the SMAC mimetic challenge. And here they also see a significant reduction in keratinocyte cell death, epidermal detachment, and lesion size. So these are some really great data, but the authors next want to understand whether their results hold up in a more human relevant context. So they test the efficacy of JAK inhibitors in a previously established humanized mouse model of TEN. Here, the PBMCs of TEN survivors is intravenously injected into immunodeficient mice. Then mice are challenged once daily with the culprit drug, the drug that induced the TEN in the patient. This induces ocular conjunctivitis, which is also seen in human TEN patients. Treatment with JAK inhibitors significantly suppressed epithelial cell death in the eye and completely eradicated disease occurrence. Pretty remarkable results that strongly suggest that this treatment will work well in human patients. And that's what they test next. They run a small clinical trial testing the efficacy of JAK inhibition in 7 patients with TEN. Notably, there was no randomization, no blinding, and no placebo control done here. There were no reported side effects from the JAK inhibitors, and 7 out of 7 patients recovered fully. This is remarkable given the prognosis of TEN that we discussed earlier. This disease is fatal in 1/3 of patients. These results are extremely promising and lay the foundation for a much larger controlled clinical trial. And that concludes the results of the paper. So let's now summarize. The authors used deep visual proteomics, or DVP, that they invented to identify many proteins in skin cell types. They recovered around 5,000 proteins per 1,000 cells for keratinocytes and CD45 positive immune cells, and around 2,000 to 3,000 proteins per 20 cells per immune cell and keratinocyte subsets. Importantly, the proteomes of these cell types were distinct in different cutaneous adverse drug reactions. One notable difference was the elevated expression of proteins involved in interferon response observed in toxic epidermal necrolysis, or TEN, compared to MPR, DRESS, and healthy controls. JAK inhibitors, which block interferon signaling, ameliorated keratinocyte killing in 3 models of TEN: an in vitro coal culture model, a SMAC mimetic-induced mouse model, and a humanized mouse model. Finally, a clinical trial demonstrated that JAK inhibitors were safe and associated with recovery in 7 TEN patients. Overall, this is a great paper. It identified a promising therapeutic target for a severe disease that lacks an effective treatment and did so using really cutting-edge technology. I learned a lot from it. The results appeared to be reproducible, as another report published by Gibson et al. 2024 in Nature Communications showed by single-cell RNA sequencing the presence of interferon-activated cell types and subsets in TEN. Going forward, I think it's important that future studies focus on better understanding the mechanisms by which JAK inhibitors are ameliorating disease in these models. As we alluded to previously, JAK inhibition will block many cytokine signaling pathways. More targeted approaches using, for example, IFNAR1 and IFN gamma receptor knockouts could help us better understand which exact cytokines are required here. I'm also very excited to see what the authors do with this DVP approach next. Will it enable discovery of new therapeutic targets in additional diseases with unmet clinical need? And can it get further developed to enable true single-cell spatial proteomics?